Today, we're embarking on a fascinating and honestly crucial deep dive into a world that's often hidden but impacts so much of our daily lives. Biosafety and biohazards, particularly within fermentation technology. To help us navigate these intricate waters, we are incredibly fortunate to have technologist Dr. Rama Yusvana with us today. Dr. Yusvana is a senior lecturer from the Faculty of Engineering Technology at Tun Hussein on University of Malaysia, UTHM. He's here to guide us through what can feel like a very dense yet absolutely vital topic. Thank you so much for joining us.
It's my pleasure to be here. Thanks for having me.
Our mission today is to truly unpack the intricacies of biohazards for you, what they are, where they typically lurk, how they can affect us, and maybe most importantly, the clever ways we manage the risks involved. It's really about understanding that invisible shield that protects us. So let's just jump right in. When someone hears biohazard, their mind might jump straight to like sci-fi movies or big outbreaks. But in the practical world of fermentation, what's the fundamental idea behind identifying these threats, and why is that idea so critical to, well, everything we consume and interact with?
That's a really great place to start. At its core, a biohazard is simply, a biological agent or maybe a condition that poses some kind of threat. And this threat isn't just to humans. It extends to animals, the environment, everything really. We're talking about things like specific bacteria, viruses, even certain fungi. The main principle is recognizing that even processes we see as beneficial, like fermentation, they carry inherent risks. Why? Because we're working with living organisms.
Right. Living things can be unpredictable.
Exactly. And for those organisms, just a slight deviation, maybe a change in the strain or an interaction we didn't expect that can turn something totally harmless into a real problem. So biosafety, especially in fermentation, it's not just some add-on, it's fundamental. It's about proactively ensuring worker safety, protecting public health, safeguarding the environment, and critically, stopping contamination of the products we're making in the places they're made. It's really the bedrock, that lets all this innovation happen safely.
That's a powerful way to put it, the bedrock of innovation. And our sources really do highlight that these biohazards aren't like a monolithic thing. They come in lots of different forms, right? Each with its own set of challenges. Can you maybe walk us through some of these different types, like from the living organisms themselves to maybe the stuff they produce? How did they present unique challenges?
Absolutely.
Yeah.
So first, we can look at the pathogenic microorganisms themselves, the actual biological agents. Take E. coli. It's a bacteria many people have heard of. Now, most strains are perfectly harmless. They live in our gut naturally. But then you have others like the notorious E. coli O157, which can be incredibly dangerous, causing severe food poisoning.
Wow, yeah.
It's a stark reminder, isn't it? Tiny genetic differences can turn something benign into a serious threat. Then you've got viruses like adenoviruses. Scientists actually cleverly re-engineer these sometimes, maybe for gene therapy. But because they're designed to deliver genetic material, they need incredibly strict handling, careful containment to make sure they only go where they're supposed to.
Makes sense.
And fungi, too. For example, Aspergillus niger is widely used in industry to make enzymes. Very useful.
Yeah.
But some related Aspergillus species, well, they produce mycotoxins, specifically something called aflatoxin, which can be highly toxic, even carcinogenic.
So it's not just about the naturally occurring threats then. It's also about what we create or modify in the lab.
It's exactly right. Beyond the natural pathogens, we have to consider genetically modified organisms, or GMOs. These might be organisms, maybe yeast or bacteria, that have been engineered specifically to boost production of something we want, maybe insulin for medicine or perhaps biofuels. The potential risk here, though, lies in their unintended spread. You don't want these modified genes getting out into wild populations.
Yeah.
Or, in very rare cases, the worry is about creating some new kind of pathogen with unexpected characteristics.
Okay.
And then there are microbial toxins. These are basically toxic substances that are produced by certain bacteria or fungi as part of their metabolism. A famous example is botulinum toxin from C. botulinum. It's actually one of the most potent neurotoxins known.
Right, but used in tiny amounts for Botox.
Precisely. In minute, controlled quantities, it has medical uses like Botox. But then you also have things like aflatoxin, which we mentioned earlier from those Aspergillus species. If that contaminates food, it's a highly carcinogenic risk.
It's really clear how living organisms and, well, the things they produce can be hazardous, but it's not just about the biology, is it? Our sources also point towards chemical hazards. What kinds of chemical dangers do we typically run into in fermentation processes, and how do they sort of fit into this whole picture?
That's a critical point, yes. Chemical hazards are definitely quite prevalent in these settings. Often, chemicals are added directly to the fermentation media. Think about antibiotics like penicillin or streptomycin. They're added to keep the culture pure, prevent unwanted bacteria from growing.
Sure.
But here's the catch. If the waste containing these antibiotics isn't disposed of properly, it can contribute to a huge problem, the rise of antibiotic-resistant bacteria. And that's a major public health concern for everyone. And we also use various solvents in these processes, maybe for extraction or cleaning. These carry their own risks: flammability, toxicity, potential respiratory issues if the ventilation isn't right or they aren't handled carefully.
Okay, so added chemicals. What about chemicals produced by the process?
Good question. What's also interesting is that the fermentation process itself can sometimes create toxic byproducts or metabolites. These aren't the things we want, but they show up. For example, some bacteria produce lactic acid. Now, lactic acid itself isn't usually a huge problem, but if it accumulates to really high levels in the culture, it can become harmful, so it needs careful monitoring. Similarly, yeast, which we usually associate with good things like bread or beer, can sometimes produce what are called fusel alcohols, things like isoamyl alcohol. These are undesirable byproducts, and they're actually toxic if consumed in significant amounts, so they have to be managed.
And it's not just the chemicals and the microbes. Our sources also flag physical hazards. What about the equipment itself? What are some of the risks there? And maybe the lab environment, too?
Absolutely. Physical hazards are a very real consideration. Take autoclaves, for instance. These are basically giant, high-pressure sterilizers. They work using extreme temperatures and pressures.
Like pressure cookers on steroids.
Exactly. So they pose risks of serious burns, even explosions if they malfunction or aren't maintained properly. Then you have centrifuges machines that spin samples incredibly fast to separate things out. If they have a mechanical failure, maybe the rotor breaks, you could have breakage, a release of biohazardous material, or even parts flying off like projectiles within the lab. Very dangerous.
Yikes.
And the bioreactors themselves, the big tanks where the fermentation actually happens, they come with risks too. Leaks, spills, potential pressure-related accidents, just because they're complex systems operating under specific conditions.
What about the general environment?
Yeah, beyond the specific equipment, the environmental conditions themselves can be hazardous. Think about labs that need very cold environments, maybe for storing samples. There's a risk of frostbite. Or places with high temperatures, perhaps near sterilization equipment, leading to burns or heat exhaustion. And critically, poor ventilation. If a lab isn't properly ventilated, hazardous substances, maybe chemical vapors or airborne microbes, can build up. That significantly increases exposure risks for anyone working there.